My name is Raoul Tibes. I'm a physician scientist here at the Mayo Clinic in Arizona, Scottsdale. I'm a physician and a researcher. I'm involved in leukemia treatment and treating patients with myelodysplastic syndrome, a pre-leukemic state. Today I will talk about SGI-110, which is a new agent that is currently undergoing a clinical testing in clinical trials for patients with AML, acute myeloid leukemia, and myelodysplastic syndrome, MDS. There are rather limited treatment options, especially for elderly patients with AML and MDS. For MDS, there have been two agents approved by the FDA for commercial use. These are called decitabine or dacogen or 5-azacitidine or Vidaza. Those are very effective agents. However, they do not work in all patients. There's a new medication developed which is called SGI-110. This is a prodrug, meaning essentially chemists and researchers were able to bind decitabine, which is the active FDA-approved drug, to another chemical modification. The idea of this modification is essentially to increase how much free drug or how much decitabine active drug eventually is released inside leukemia and MDS cells, while at the same time attempting to avoid some of the side effects and toxicities of these known agents. So the study is a phase 1-2 dose escalation study of SGI-110 that took place in several cancer centers in the United States, including here at Mayo Clinic in Arizona. The goal of the study was to see if this new formulation of decitabine can achieve as high and as good levels of the drug inside the blood, as well as even higher levels inside leukemia and MDS cells. The idea being then that this new medication, SGI-110, would be more active and more potent against leukemia and MDS cells. The primary goals, how we call it, in the trial was not to test the activity, however, but it was more to see if the scientific rationale how this medication was developed is actually true and holds up. So essentially, can we prolong the active medication inside the bloodstream? SGI-110 is able to lead to alterations and re-expression of some of the genes to a higher degree than decitabine was able to do, or I should say at this point to a similar degree. However, the drug clearly had a longer half-life, how we call it, which is indicative it sticks around in the blood longer. So far, in preliminary responses, we already have seen complete remissions in a few patients that were treated on the clinical trial. Mostly in those patients that had acute myeloid leukemia or AML. In several of those patients, these patients have received multiple other therapies, including a stem cell transplantation. I think this is quite encouraging to see responses in that patient population, specifically with patients with acute myeloid leukemia that have received several other treatments before. In patients with MDS, I think it's important to mention that, at least in those preliminary results as presented today, in patients that have received and have not responded to prior decitabine or 5-azacitidine, the other two drugs that are approved already, we did not see too many responses. However, we need to further investigate as to the activity of SGI-110 in myelodysplastic syndrome patients. These are early clinical trials results from a phase 1 trial. Further research is needed in both MDS and AML patients to determine which are the patients that may respond, which is the appropriate patient population, and it's still a long way ahead of us to find out and figure out what are the best patients to treat and if this novel agent is truly better than the already available agents. However, it is promising and is one of the only new agents out there working through a similar mechanism of two good drugs we already have in the clinic.